session, and I hope you guys had an interesting session last week, and you would have had a chance to go through the online modules as well. So before we actually start up with today's session, I would like to go through the organization page um, that is being set up for this particular program. It's okay. I guess my voice is echoing. Okay, I'll look into that apart later. So yeah, this is a kind of a personalized dashboard.
Hi,
Okay.
Sahil. So I think it's important to let people know that they need to mute themselves.
Uh-huh. Yeah, I did. Okay, write it down on the chat as well. So please, it's a request from all the participants to mute your mics as soon as you join the meeting for the better experience. Okay, looks like it's better now. So let me restart it. So yeah, I was about to go through the organization page or the dashboard that we have created for this training program. So I'll just walk you through what exactly it is and how exactly it is beneficial. So over here, that's the link. I can type down the link on the chat. So we have different tabs over here. The first one that to start with is the home tab. So this particular tabs keep you updated with all the things that we are posting. So like, we have the files and the presentation that is to be used in today's session or the recording of the previous session. So you could see all the updates listed down in the home tab. And as we mentioned in the last meeting that we have forums as well. So if you feel like putting up your questions over there and you want other participants to reply on that or to interact with other participants to get a better experience of what the other participant is doing, so you could create the forum. Otherwise, we are going to create the forum topics for all the recordings and the files that were used in the session. So you could go back and track down all the recordings and the files. Now the other thing that is pretty important, that is the progress. So all the participants could track their own progress, like what exactly have you been doing on the educational side and on the TBio info platform. So I'll just show you how exactly this works and how exactly you could analyze it. Um, this is the progress bar that is being shown for this current week. You have the timeline that you could select, the time range that you could select where you want to see your progress. So let's say for this particular discussion, I'm gonna choose the start date of 10th of March till 23rd of March, and I'm gonna go and click on apply. So now this particular time range has been applied and I could see all the progress that has been made. And then over here we have the points. So for each and every activity that is being done by the participant on the educational side and on the server can be seen over here. So and moreover, it is very essential for all the participants to go back and check out our online modules soon after the session so that you are up to the mark and you understand what exactly was done during the lecture. So, for example, the previous lecture that we did, could be correlated with what we have on Introduction to Bioinformatics. So you can just go ahead and take the course, and all the courses are free for all the participants. So you could get a better understanding what exactly was covered in the previous section. And I would really like to call off Phil's name. Okay.
एक एक मिनट में जब अपन म्यूट को दबाएंगे तब रेड रेड कलर का हो जाएगा ना वो।
Mute yourself.
हाँ मुझे पता है कैसे
उसके बाद ही नहीं होगा देख वीडियो वाला।
I guess.
अरे मुझे ये बता जब अपन उस रेड को दबाएंगे तभी म्यूट होगा ना मतलब
I'm sorry about that. So yeah, I would like to call off Akhil's name, Akhil Rajput. He has been pretty active with all the activities that he's going through. So just to have more information, like how exactly the other participants are doing, we have a small click on button of information over here. So I could get more information about like what exactly. Okay, yeah, I guess I have some network issues. So you can have like a personalized information when you click on the information button. So that's more about the progress. And the last thing is I would really like to thank everyone for taking out the time to fill out the survey form that was circulated last week. And thank you for letting us know about your educational background and the projects that you guys are interested to work upon. That was really insightful, and we will make sure to reach out to each and every participant and try to know their interest and their project ideas so that we could start getting ready for the project, and you could have a more clear understanding when we will be going through the data analysis steps on the TBioInfo platform. So I would really thank you guys for filling up the survey form and letting us know more details about yourself. Now, without wasting more time, I would like to welcome Mr. Ilya Brodsky to continue with today's presentation.
Thank you, Sahil. And again, yes, as we can hear the sounds for people that are joining, if you could please mute yourself, and Sahil, maybe you can make sure that people that recently joined, their microphones are off.
Sure.
And anyone that wants to say anything, there's an option here to raise your hand. That might be a useful way to get attention to whatever you want to say, or if you want to just mention something, please put it in the chat. So thank you, Sahil, for giving a little bit of an introduction to how things work. I do want to mention that on the survey results, it seems like a lot of you have been actively publishing, and also you plan to utilize the skills acquired in this training program to advance your research. So this is very good. We'll try to be in contact personally with some of you that are interested. We know already from the first survey which projects seem to be more interesting. And so as soon as we get to the point of knowing how to process the data, knowing how to find the data that is interesting to process, and then downloading it and making sure that it's all available, we'll be transitioning into that phase of working on independent projects. So today's session is focused on processing of next generation sequencing data. Sahil, it seems like someone else joined, so we can again hear. Okay, so please, if you're just joining, please mute your microphone. That will help everybody else hear much clearer. Thank you. So today we want to talk about processing of next generation sequencing data. And we want to talk about it in two ways. The first is the role of pre-processing. Sahil, can you please mute whoever has their microphone on?
Yes, I did that.
Oh, I can still hear someone. In fact, I can see that it's Bowtie, probably, and there's a few people. So the role of pre-processing is important to understand because it is related to the technology of next-generation sequencing. And then the second big portion of processing next-generation sequencing data is going to be focused on mapping techniques. So we'll talk about several different approaches to mapping. Before we start, let's talk about how did we start with next generation sequencing data and how we can utilize this data to do research of biological mechanisms and what kinds of biological mechanisms we can study with next generation sequencing data. You're probably familiar with this, but just to give you an overview, genomic data is something that has been available only recently. And how did we get to this point? In the 1950s, the DNA structure was discovered. And then very soon after that, everybody was aware of the DNA and the role it plays in molecular biology. And in the 1990s, the Human Genome Project kind of brought that front and center for everybody to be aware of the revolution that this type of data could bring to our understanding of human health and disease. And so the Human Genome Project really opened up a lot of those data sets, but it was a transition from a standard sequencing method to a new sequencing method that eventually was popularized by Illumina. And so a lot of times when we say next generation sequencing today, we will be referring to Illumina sequencing, even though other methods are available. And so Illumina really made it much more affordable, but their methodology needs to be understood so that we can properly address the processing of next generation sequencing data. So in 2006, Illumina launched their first sequencer, and as a result, you can see that the time kind of accelerates from there. In 2015, we already had 1,000 genomes that were available, and very soon after that, in 2018, now we have access to over 100,000 genomes that were sequenced and that are being used for research, but also in reference to health and disease and biomedical purposes. So in terms of how quickly we got to the data about many different organisms, but a lot of it is about the human genome, of course, has been very fast and really got accelerated because of this next generation sequencing technology. So today we want to talk a little bit more about what that technology really does, how do we get that sequencing data, and then we want to talk about how to address the preparation techniques that next generation sequencing uses to produce the data, as well as how to map on these different genomes that are available to us in different repositories like NCBI. Now, Justin, I want to turn it over to you so that you can say a few words about next generation sequencing in terms of how it's used and how it's changed research. Justin. So next generation sequencing, I'll just say a few words about it, really has transformed how research is done in the lab.
Yeah,
because, okay, can you?
I'm here, sorry,
okay,
yeah. Technical issues. Greetings, everyone. I'm Justin. And before Ilya goes into some of the more technical aspects of next generation sequencing, I wanted to just make a couple points as a neurobiologist who has an awareness and appreciation of the technology. And I guess my first point, I know we have a lot of biologists and biochemists here, is NGS is not really limited and is not used exclusively by biology. It really is applicable to many different areas. And one of the main reasons for that is as it has developed, it not only generates lots of information and can do so very quickly, but we are now, sorry, some static. Now we're able to take all that information and interpret it and analyze it. So in the beginning, we were generating lots and lots of sequences, but that doesn't do a whole lot of good if you can't make sense of it. So my last point, and of course we could talk a lot about clinical applications and many others, is that this ability to sequence very quickly and accurately with precision now allows us to make very clear comparisons and accurate comparisons between genomes. And that's obviously invaluable when we're trying to conclude that one mutation or a sequence is actually causative or involved with some, for example, neurobiological disorder. So it's really exciting. And for those of you who don't have a strong awareness at this point, you should over the next 30 minutes. So thanks for listening, and I'll turn it back to Ilya.
All right, thank you, Justin. Yeah, so it's really something that is important in terms of impact, and it already can be seen, I think, in the lecture today, how the availability of data, the affordability of this kind of data can play a major role in how we conduct research. So first of all, where is this data, right? So that's one of the things that we want to touch upon. We're going to be working on public domain data sets. And so I want to just briefly talk about where do you find a lot of this data. So there are several things that we need to understand about the data that is available on places like NCBI. A lot of the publications that you will be reading are going to include references to a published data set that was used to produce the results that we discuss in the publication. And in a lot of the publications, if you just look for SRR, right, that SRR stands for Sequence Read Archive Repository. So this is going to lead you to NCBI, the SRA. And the SRA includes bioprojects where you can download the data sets. And so at some point during the presentation, I want to give you a little bit of an overview of how you get that kind of data and how you can utilize it throughout this program as well. But this is just a little preview. So a lot of that data has been reposited with NCBI and is available for us to reanalyze, and the data sets that we'll be using, they all come from NCBI, the SRA repository. So what is the SRA format, and what is included, and what kinds of NGS data formats are there? First of all, the Illumina sequencer will produce two types of formats, and we'll kind of talk exclusively about Illumina, even though there are some other ones, and there is some overlap there. So you will hear formats like the BAM format, the SAM format, but all of them are derivatives or could be converted to some of these formats that we have in front of us, the FASTQ and the FASTA. So what is the FASTA format and the FASTQ formats that we will be looking at? FASTA format, Sahil, if you could please mute the people that are joining, that would be very helpful. So the FASTA format is simply sequences, raw sequences of reads that are produced through the next generation sequencing technology. And those will be short reads, as you can see right here, short sequences that range anywhere between 35 and 215 base pairs in their length. And as they come, for example, in a reference genome, it could be a complete long sequence, or it could be short sequence reads, just like you see right here. The FASTQ format is essentially a very similar format where you have a sequence, but you also have information about the quality of that sequence. So for example, the FASTA file will be typically where you store a reference genome. So if you map onto something, you're mapping on a reference genome, and that reference genome will be in FASTA format typically. And so you can see how it's just a continuous sequence with the reference of which organism that is. And typically that organism will also have a code, an NCBI code, something like this, and then a description of what it is. So that is on the FASTA format. What about the FASTQ format? The FASTQ format actually contains a lot of the raw data that comes from the sequencer itself. So when we talk about next generation sequencing, the way that data is prepared is that DNA is sheared. So you can see that it's cut into smaller pieces. between 200 and 300 base pairs long. Then adapters are attached to those sequences. And then in flow cells, those elements are amplified. So they are amplified so that they could be read by the sequencer. And the sequencer essentially detects letters based on colors. And so those colors have a quality of signal. The more clear the color, the more reliable is that signal. And so you can see that the FASTQ file contains that information. It will contain the sequence that the sequencer read from the DNA. And then you also have a score that will be associated with each one of those letters. Also, we will look today at how we have two types of sequencing. We have single-end sequences and pair-end sequences. And so single-end would be sequences that are just read from one side, and paired-end would be read, the same sequence is read from both sides. That technology is considered to be more reliable because now you have the same sequence read twice. So if everything is consistent, then you're probably looking at a very reliable read of that sequence. However, those individual reads, the one that is forward strand and backward strand, are going to be, or forward direction and backward direction, sorry, those are going to be placed separately. So you do have to combine them together before mapping. Next generation sequencing is used for many different reasons. And so if we look at kind of this overview of data from phenotypic data, what we observe, for example, cell culture or tissue images or some kind of phenotypic image or phenotypic observation, it could be clinical data, any other kind of data, that could be linked to, for example, genomic data, right? So what is genomic data? Those sequences will have individual letters that are in the read that we are looking in and the reference. And so by looking at the difference between the read and the reference, we can identify several different variations. For example, substitution, insertion, deletion. And so we can look at a variety of changes in the sequence. Then we can also look at various epigenetic signals. So epigenetics has been a growing field in its popularity. And epigenetic data is essentially the same next generation sequencing data, but instead of looking at the changes in the DNA, in the code that we have, we are looking at histone, those areas of the DNA that are wrapped around the histone, so they are not read easily. And for example, DNA methylation. So where we have CpG islands and methylated sections that are also not available for reading. So those are the epigenetic type of variations that we can observe and learn through next generation sequencing data. And also we will look at transcriptomics, so how we can measure the expression. And of course, all of these are linked together, right? So we do want to study how changes in the phenotype are explained through the various types of data that are contained in the DNA or in the transcription process from DNA to RNA. And we can also go on, and this is now not related to next generation sequencing data, but we can also look at the product, the proteins, the metabolites that are the product of mRNA being translated into proteins. So next generation sequencing in itself covers a large portion of that whole process. And we can use it by processing next generation sequencing data that was prepared with different types of preparation kits. And then we can measure the signal. And sometimes if we have signal at multiple levels, integrate those signals to get a real systems biology perspective on what's going on and the underlying mechanisms behind the phenotype that we observe. So what are some of those processing tools that we have available and we'll touch on throughout this session? RNA-seq, where we can study the expression. But when we talk about the expression, we're talking about gene expression, but we're also talking about isoform expression. So how alternative splicing can be observed, and how do we quantify it so that we can measure, for example, differential isoform or gene expression, and how we can study all kinds of rearrangements within those isoforms. We can also use ChIP-seq or chromatin immunoprecipitation that we can analyze interaction between a certain target protein, for example, histones, or different components of histones, and how we can then study peak calling and understand epigenetic variation related to histone modification. There's also microarray technology, but also this applies to the sequencing of whole exome or whole genome sequencing. So we can study the actual sequence of the DNA. And then, like we said, we can also look at RNA editing events, various repeats, rearrangements, and other things that happen to RNA. So there's a lot that we can do with next generation sequencing data. And now let's take a look at how that actually translates into the data analysis side of things. So on the platform that we looked at a little bit last time, we have a whole dedicated section to next generation sequencing data that covers things like RNA-seq, which would be the first one right here. You have RNA-seq of single cell, RNA editing, and then under genomics and epigenetics, we have whole exome sequencing or whole genome sequencing, where you can detect mutation variants. We have bisulfite DNA methylation analysis, which is whole genome or targeted methylation, where you can identify methylated regions of DNA. ChIP-seq that we talked about just recently, copy number variation, and we have a whole section related to other types of sequencing for DNA and RNA. For example, unsupervised analysis of non-coding RNA, repeats, genome segmentation, etc., microRNAs, and metagenomics, microbiome, and also amplicon 16S, which is also related to metagenomics. So today we want to talk about some of these methods in processing this kind of data. Now, as you remember, we talked last time about how all of these sections have a consistent interface, and this consistent interface will help us transition between the different sections fairly easily. So I'll just go over this really quickly, and then we'll jump onto the platform so that you can see how we can take a raw sequence and visualize what are those reads in terms of their mapping on a reference genome. So you can, if you go in, let me switch over to my computer screen. So if you go into your account, and please let us know if you have any issues logging in. Once you're logged in, you should see your email address right here. And I think by now, all of you should be kind of familiar with the interface. If you have any questions, you can either ask today, or you can also post them here in the chat box. As we saw, we have this next generation sequencing section right here, and let's go into the RNA-seq section so we can kind of see practically how this is done. So first of all, in every section on top, you will have this selection of the different data formats and the different presets that we have for starting your pipeline. We have here a variety of either model genomes that have a good annotation and are already preloaded on the system. This is our model genome GTF. We have an option for when you can upload your own genome. For example, download a genome from NCBI and upload it onto the server, and then you can map things to that genome. Or sometimes you will have a genome that comes with a GTF file. So that is something that you can see, for example, on NCBI. GTF or GFF is a reference file that annotates the starting and the ending position of genes and exons that you want to use in your reference genome. Here we talked about several of those raw input data types. We have FASTQ, which is what we talked about. It has the quality score and could be either single end or pair end. FASTA, which is just reads without any information on the quality. SAM and BAM files. And then also sometimes you can use simulated files if you would like to check how different methods work or compare to each other. Some of the organisms that have been, by organisms I mean genomes of organisms that are well annotated. So if you use our model genome GTF, you can utilize some of these preloaded organisms. We have Arabidopsis, 2 assemblies for human, mouse, etc., and human-mouse concatenated. This is something that we'll be using in one of our projects. And finally, here you can select single or pair end reads. Once this is set up, you can go ahead and upload files and groups. And so when we do this, I just want to explain exactly how we will do this. So obviously, you can select, for example, the human genome and single end, and this is just a demonstration. You don't have to think. In a little bit, we'll be creating a pipeline together, so you'll be able to use real files, but I just want to first go through the steps. Once you click on upload data, you will be able to do a variety of things. You can upload data that you've downloaded from NCBI, for example, and that will be FASTQ files like we just selected. You can also use reference links, so files that are already in the repository on the system, and the majority of the data that we'll utilize throughout this program in the beginning will be data that is already on the repository. As a part of this program, you right now have a limit on upload. So if you want to upload a specific data set, you have to talk to us first, tell us about the data set. We will make it available for you. It will be on the repository, and then you'll be able to utilize it. And then you have this option for reference links. So I'll just explain how that works. Let's say I have a data set that is called ilia.fq. Okay, let's say I have some file that's called ilia.fq, and I have another file that is called sahil.fq. So FASTQ files, and I want to compare one against the other. So now I can see that those files have been properly configured. And for example, I can compare them by dragging them into 2 separate groups, and then I can analyze the data using different types of contrasts. So we'll talk more about this later. Right now, this first portion of the pipeline has been prepared. So we have everything selected for the right type of upload, and we are able to now build our pipeline. So once you see that you have the start option available, and now you can see that some sections are darker, some are lighter, which means that you can start using these sections. The first thing that you'll notice is that some of these are organized into groups. These are logical steps. So for example, pre-processing, error correction, mapping on genome, exon detection, etc. You can see for yourself. And by selecting any kind of a button, you will see that it's connected. So later on, you can understand what you did to get the result that you were looking at. So if I want to go back, I just press the right button, and now it goes back to the previous step. So this is a little bit of an introduction on how the platform is used. Once you create a pipeline, you will see under My Pipelines a number of different pipelines that you've run, and you can go inside the pipeline and see what you did. And sometimes you will have outputs right here. Sometimes you will have visualization outputs, and you can. We'll talk more about the different formats of outputs that will be useful for a session. The first pipeline that we want to prepare today is going to deal with mapping the reads, the raw reads and FASTQ files onto a reference genome. So again, just like I was showing, we have an SRA file. So this is a file downloaded from NCBI, and SRA, all it does is it compresses the raw FASTQ file. And then we have a genome, a reference genome. So this is something that was taken from a publicly available data set based on production of biologics, so production of vaccines, a cell line that was used in the production of vaccines. And we want to see how can we map those reads onto the reference genome. So let's do the following. You all should have this presentation. So what we want to do is we want to copy this first link, okay, and create an RNA-seq pipeline. And I'll just kind of walk you through the steps. We have here our genome because we're just going to be using a FASTA file of a new genome that is from the SF9 from Spotoptera frugiperida, which is an insect cell line for the production of biologics. It's going to be a FASTQ file, even though it's SRA. Inside the SRA is a FASTQ file. And we are going to use single end reads. Now here we paste that first link. So this is going to be gene expression from this cell line. Once I pasted this link in here, I can also upload my organism. So I need to take the second link and paste it in here. So now I have my FASTQ file and I have my reference genome. I don't need any groups because all I want to do at this moment is visualize the reads, how they fall onto the reference genome. So I don't need any groups. And now I want to start. Here you can see a little bit about an overview of what RNA-seq technology is. And I want to use Bowtie to map those reads onto the reference genome. Now, what is the problem with mapping short reads onto the reference genome? The problem is that computationally speaking, this is a very intense procedure. You have to take millions of reads and you have to put them onto a reference genome that is also several million nucleotides long. So this is computationally a challenging task. And Bowtie is one of the methods that could do this. Now we are using Bowtie 2G. So we are using a reference genome and not a transcriptome, right? You can see mapping on genome, and we are using Bowtie 2G. The next thing that I want to do is just visualize my reads. So we will use JBrowse, a visualization solution, a publicly available solution that uses BAM files that will contain the information about where those reads fall on the reference genome. So this is using JBrowse. And again, remember that the input into this visualization module is going to be a BAM file. BAM file stands for binary alignment mapping. So BAM file, visualization, and now we click on end. I can save, for example, SF9 visualization after Bowtie 2G. Once I save, I have to remember to click on Run Pipeline. So once I run the pipeline, I will see that it reloads, and I should see that this is started. Okay, now you don't need to keep this window open. You are submitting a job onto our cloud infrastructure. So once it is complete, you will get a notification by email, or you will also see it right here under My Pipelines. So you can see that right now my pipeline is processing. If I reload, I will see how it updates. But it will take some time because I took a full FASTQ file that's about 2 gigabytes in size, and I map it on the genome of this organism. So now you can see that this is the pipeline that we built, right? So we mapped our raw FASTQ file onto a reference genome, and the result will be a visual output. So let's quickly take a look at what this output looks like. I am giving you here, again, you should have the PDF of this presentation, so you can see the link. It's right here. If I copy this link, and all of you can copy the same link and you go there, or you can always go to pipelines / and then this is a unique identifier of your pipeline. And you'll be able to see the result of my pipeline. So this is a pipeline that has completed. You can see that it's 100% done. And now let's take a look. Over here, it has a button that is called visualization. So what you will see is something that looks like that. If you click on that button, again, if you go into the presentation, you can also take this long link, and that will take you to that output as well. What are we seeing here? So you can see here that we have several tracks, right? So several layers of information. On the very top, we have the reference genome. It's about 4 million nucleotides long. And we are looking at this small section that ranges between about 1.9 million to about 2 million. So right in the middle, we are looking at this small portion of the genome. Over here, we have, as you can see, coverage and SNPs. So coverage means how many reads fall onto that segment, okay? And you can see over here the scale. The scale goes from 0 to about 400. And then underneath, you can actually see the alignment of those reads. And you can see here the red and the blue. Those are forward and reverse directions that we use paired end reads from. So now we can zoom in, okay, and we can see a little bit more detail. For example, you're starting to see those single nucleotide polymorphisms. So you can start seeing how in specific places there's a change, okay? And how do we know what change it is? We need to zoom in even more so we can actually see the sequence. Zoom in a little bit more, okay? And now you can see what those changes are. So for example, here, you can see that this is a G, and it happens in some portion of the reads, not all of them, but in some. Okay, so as you scroll, manually you can just navigate, and you will see the points over here that indicate a consistent change. So sometimes you will have change that doesn't occur in all of the reads. Sometimes you will see a change that occurs in all of the reads. So this is just a way to visualize how those individual reads map onto the reference genome. So this is the reference genome. What is coverage? And again, coverage is just how many reads do you have aligned to a specific segment. You can see that this scale changes as we go between low coverage and high coverage. So you can see that we will get to a region that now has a lot of coverage. What does this mean that the coverage is higher? Because this is an RNA-seq file, actually more coverage means that that portion is expressed, okay, is more available. So more mRNA was available that aligns to that segment. So if we zoom out, now we can see that over here is a portion that is expressed. So this is more expression than, for example, right here. And some regions will have nothing. But of course, this is mapping onto the full genome. So we're including here both the exons and the introns. And so we are just visualizing right now what does it mean that we align the reads to the reference genome. Okay. So now if we continue, let's understand how we can use this information and what other steps are necessary to be considered when we talk about RNA-seq. Well, one of the things that we already talked about is pre-processing. Pre-processing is essential for us to get an accurate readout of the signal that comes from a next-generation sequencer. Why? Because this is a quick overview of how that process happens. We have the recorded genes. Those genes are on the DNA, and some of those genes are transcribed into transcripts. So for example, out of these 3 genes, gene A and gene C get transformed into transcripts. And so we have maybe a different number of transcripts of A and transcript of C. These transcripts become cDNA and they're shattered. So they're shattered into small portions between 250 and 200 to 300 nucleotides long. And adapters are attached to those sequences, to those short sequences. So at the end of each one of those fragments, we have a beginning and an end adapter. Then these are amplified. So the amount of reads is amplified so that the reading of the material is more clear. And that's when the reading happens. So when we read, right, the actual reads that we are looking at in FASTQ files, they include the adapters and they include PCR amplification. How can we see PCR amplification? If we go back here, you will notice that some regions have this strange kind of continuous replication of the same read. Sometimes this means that this is PCR amplification. This is not representative of biological variation. This is just a technical variation related to the library prep, right? So it's related to how the data is prepared and then sequenced. Because of that, we want to eliminate these kinds of amplified regions and make sure that we go back to what was in the original sample. The second thing that we cannot detect here in the reads, because the reads is what we got as an output, is those adapters. So adapters actually interfere with mapping because they're not contained in the reference genome. So how do we deal with these technical issues? Let's go back to the platform real quick and create another RNA-seq pipeline. You can see that these pre-processing steps are here to help us eliminate that technical variation. So you can see PCR-clean and Trimmomatic. Those are 2 of the methods that we'll be using to trim the adapters with Trimmomatic and remove those areas with PCR-clean. So before we move into mapping, we have to sometimes address the issue of pre-processing. Now, how do you know whether you have to use it or you don't have to use it? In a lot of the methods, when you read the publication that describes the methodology, you will also find that they tell you what did they deposit in the sequence read archive. Sometimes it will say that they just dumped all of their material in there. Sometimes they will say that they actually already cleaned out the adapters and PCR amplification. Okay, let's take a look at this example really quickly. Where do we find this information? So the publication that we have and this first alignment data set that we were looking at comes from this publication. If you just search here for SRR, you will see that they explain where their data is, right? So they say here how many reads, in total 48 million raw reads, but they did clean them. So you can see that 48 million reads was transformed into 47.5 clean reads after removing reads containing adapters, reads containing poly-N, and low quality reads. Right? So they already did some pre-processing before depositing the clean reads into the SRA database, and they give you the accession number. So how do we find this accession number? We can go to NCBI, select here SRA, right here is the sequence read archive, and input this number right here. So as a result, we get that the raw data that they were describing, right? And here you find that information that will be useful for our analysis. For example, we see that this is RNA-seq, and this is Illumina data, and this is paired end read, right? So all of those inputs that we started with in the beginning, typically they're going to be given right here. Unfortunately, not always, and sometimes you need to guess, or you need to know by the files that you will download. Now, how do I get this raw file? I can go over here to the run selector, and I can see all the information that there is about this, right? So you can see that all the same information is right here. And you have several different links right here related to the whole bio project or which organism this is, etc. So, but if I click right here, let's wait until it opens. Okay, so here you have access to the raw files, right? You can see right here. So one of the things that you can do, if you know how to use the SRA toolkit, you will be able to get the raw FASTQ file. Okay, so you will have the metadata, or you can download the raw FASTQ file. But a lot of times, for example, in our case, right, these are huge files. So you can see that this is going to be a 2.6 gigabyte file. And it's sometimes, difficult to download all of that, unpack it, and then upload it back on the system. So this is going to take a long time. So I'll just show you a shortcut that you can do right from here. If you download the run info file, it will actually generate a downloadable CSV file for you. And if you open this file in something like Excel or a text editor, it will actually give you a direct link. So now you can see the HTTPS link right here. And this link is directly to the SRR. Okay? Don't try to use this link on the platform itself, but whenever you're preparing your project and you plan your analysis, include this link in your analysis plan. That way we can pre-upload it on the repository and make it available for you. Unfortunately, just like this, it is not going to work because it is not a direct link to the SRA file. It's just a link to the folder where the SRA file is. So we'll have to still upload it onto the repository. So this is a little overview of where this data came from and how to know whether this is clean data or we need to do some pre-processing. The other thing that we want to look at, sorry, let me go back, is what is the impact of pre-processing steps? So the pre-processing steps are going to be most important for highly expressed data points. For example, here you can see a comparison without pre-processing and with pre-processing. What do these charts tell you? These are the transformed, so log transformed FPKM plus 1 without any duplicates, okay? We are looking at how do these two samples correlate in terms of their genes, right? So this is sample 1, this is sample 2, and you can see that if we don't do pre-processing, we will have a deviation, right? And so what does this mean? This means that for highly expressed genes, these are the highly expressed genes, we will see a biased view of their expression. So in some cases, when these files are not pre-processed, it's important to pre-process them, and then we will get an accurate measurement of expression of highly expressed genes. Okay, now let's talk a little bit more about alignment. We already talked about Bowtie 2G and alignment of reads to the reference genome. In reality, we need to understand a little bit more of the various types of reads so that we know how to select an appropriate method of alignment based on what we need to get out. So for the first one, when we just want to visualize the reads, Bowtie 2G is the way to go. But what goes into alignment? So whenever we have a sequence, for example, this is our reference genome, and this is an output of the BAM file. So this is what's inside the BAM file that, for example, Bowtie 2G produces. It will contain these sequences. These are sequences of reads, and you can see that they are organized by the same beginning. So that first step in a lot of these mapping techniques is called indexing. Indexing means we organize reads into groups by their beginning. So we try to align them. All of these begin with TCCGA, we put them together. These begin with CCGA, we put that next. This with GAGA, we put it next, right? So this is called indexing. And then based on this index, we can align them much faster to the reference genome. The other thing is we, of course, talked about the single end and the paired end reads. And here you can see how those genes would be, or how those reads would be aligned to exonic regions, and then how we can use the paired end alignment so that we better understand what is expressed and what is not expressed. And so as a result of this, you can see that one of the challenges will be quantification. Because what do we need to quantify? We need to quantify full sequences of genes from here to here. So given different alignment of reads and different alignment of those reads to the different regions, we will actually be getting different results. And so one of the challenges is going to be quantification. So to get an overview of the major steps involved in the RNA-seq processing of data, these are some of those steps and some of those challenges that we first need to address. Pre-processing, removing adapters and trimming, removing PCR duplicates, and you will see that this is color-coded on the platform in the color blue. The second step is mapping, where we can map on known sets of transcripts, for example, known isoforms. We can map on the genome, and we talked about how we do that with Bowtie 2, or a combined strategy. And so we'll talk a little bit about some of those methods like TopHat and HiSat that allow us to combine that strategy. And then the final challenge is going to be quantification of expression levels. How do we then take the alignment of reads and say this gene or this transcript is 100 or 200 expression, right? And what is that expression and what values are we talking about here? So to explore how to do this, let us create two pipelines together. And I'll tell you a little bit about the data that we'll use to do this. So the first pipeline is going to be very simple. We're going to do the pre-processing steps. Then we will do alignment or mapping on transcripts. So these are going to be known transcripts. And then we will use RSEM, or expectation maximization, a statistical technique to understand where those reads should be and how do we quantify them. And the second pipeline is going to be a little bit more expanded. So this is going to be your standard tuxedo pipeline, where you can see how we will use pre-processing, then a combined technique where we will map on the genome and identify junctions and exons, construct isoforms, and then map all of our reads on newly constructed transcripts and quantify those transcripts and what we will get at the end. Now, before this, you received a file, and I believe that Sahil pasted this information on this page. So if you haven't downloaded the files yet, please go over here and you can see that we have a link to the files. Let's see what's inside these files. Inside these files, we have samples.svl. That's the file that we want to use. And then we'll see what those samples are in this Excel sheet. So those are the names, the phenotypic information about the data that we're using. So let's first take samples.svl and understand what is inside this data set. So samples.svl looks like this. It's just a link to a bunch of different files. As you can see, each file is mentioned twice. So you have SRR 25697, SRR 2925697. But at the end, they have _1 and _2. These are paired end reads. So when you see _1 and _2, that tells you already that these are paired end reads. And you can see that there's a number of files all together in a single reference link. So let's quickly create this pipeline. So first of all, we know that these are paired end reads, FASTQ files, and this is human data. So these are human cell lines. We will use the assembly GRCH38. Upload files and load groups. We will go to URL bulk upload. That is when we have this SVL link. We will use URL bulk upload. Now under let me just quickly find that file. Samples.svl. I choose it. Define groups check. Now you can see that it correctly identified that these are paired end reads. I don't need to create groups. And now I'm going to run two pipelines. The first one will use Trimmomatic PCR-clean. Then I will go to Bowtie 2T, RSEM expression table, and here you can read about how quantification is done in different measurements. We'll talk about this in our next session. And end. So this is a pipeline that uses a simple mapping on known transcripts for the cell line project. Save and run pipeline. And once you do run pipeline, you should see that it has started. Go back to my pipelines and you should see it in green. So this is a little overview. We're running a little bit out of time, but this is a little overview of how do we start analyzing the data, and this is the pipeline that we just ran together. How do we start preparing and processing these short reads, and how do we map them onto the genome? What does that mean? And how can we quantify the output? And so next time, we'll talk about quantification. And please, whenever you are preparing for the next session, you can try and go through Transcriptomics 1. In Transcriptomics 1, we will talk about some of these methods, and you will have a step-by-step tutorial on how to do all of these steps and what they actually mean. So just to show you where that is, you can go to, let me go back. If you go over here under courses, you can see Transcriptomics 1. Transcriptomics 1 will give you files and a step-by-step tutorial on how to go through the steps that we just did today. So if you complete this session by our next time, you will have enough practice and enough experience working with raw FASTQ files. Tomorrow, or in the next time that we will talk, we will touch on Transcriptomics 2. And Transcriptomics 2 will talk about different quantification techniques, including differential gene expression and various adaptations of the regression model to study variation in RNA-seq data, in gene expression data. Now, I would like to conclude by saying that we did invite all of you to participate in our computational neurobiology workshop that will be at the coming up Louisiana State University, LSU, Center for Computation and Technology. They will host the annual bioinformatics conference, and this workshop will be right before the conference at the same place, at the CCT, the Center for Computation and Technology. We all, I think you all should be getting this link, especially if you are in Louisiana. And this is just, again, something that you should also consider. The upcoming conference, somebody from our group will be presenting, but also many other distinguished speakers from around the US. So if you're able to come, you can register. And so just to show you a couple of those links, this is the link to register for the workshop. So if you scroll down right here, you can register right here at the bottom. We will also have remote access. So if you're around during that time, we'll be able to send you the details for remote access. And over here on the last slide, you can see LBRN conference where you have to register as well. So if you want to participate, please register there as well. That's all I had for today. If you have any questions, we would be happy to talk and help you out with any technical difficulties you might have. I hope that the session kind of explained some of the pre-processing steps and the mapping techniques that are used. We'll try and cover more in our next session. And please make sure that you look at the online materials that we have so that everybody's on the same page by the next time. And like Sahil showed in the beginning, you do have the ability to kind of monitor the progress for yourself and compare it to the progress of the whole group and kind of see where everybody's at. Thank you, everyone.